Hi, welcome to this lecture on environmental adaptation of animals. On these videos, we will consider a little bit on energy metabolism. So we already know what kind of nutritions we have and how much energy we get from fat or proteins or sugars. And now we might think that, okay, how we can consume them and where do we consume it? Let's start again with polar expeditions. In the previous video, I showed the Scott expedition in beginning of 20th century. And it was a catastrophe. They didn't have enough food, so they starved to death. And in the mid-80s, people were already knowing that, okay, how much energy there exists in fat or proteins, etc., and how much energy they need for these kind of expeditions. But still, this is an expedition for North Pole, and they need 630 kilograms, mainly food, for 2 persons and the dogs. And so even then, they need a lot of food, and although the food is mainly fat, so it's more calories per gram than other nutritions. So where do we need the food at all? Because we are adults, we are not growing anymore larger. And we can blame the thermodynamics. If we have water that is flowing in isolated system, it will release the heat on the surrounding. So in the beginning, the water molecules have much energy in directional movement, so they are flowing fast. But then later, they will flow slower, because now there is a random motion in the water particles, random motion of copper atoms on these tube walls. And this is caused by the second law of thermodynamics, saying that, okay, if there is a change, the direction of change is always toward greater disorder. And this works in isolated systems, and nature is not isolated. So that's why we require some energy from outside all the time to create and maintain the essential organization, so to maintain the certain ions outside and certain ions inside and make new proteins, etc. In the human body or any other animal body, there is a different kind of energy, and they can have different kind of usefulness. First of all, we have chemical energy, meaning in the chemical bonds. And a good example of these is, for example, ATP molecule, from which we can cleave the last phosphate and release energy. Then we have electrical energy, meaning that, okay, we have separated positive and negative charges. We have these ion pumps that are moving sodium out from the cell, etc. And we can use these for controlling different cells, for example. Then there is a molecular kinetic energy, meaning heat. So there is a random atomic molecule or molecular motion. And last but not least, we have mechanical energy. So the molecules are moving in the same directions, or the cells are moving in the same directions, or the whole animal is moving in the same direction. So it's a moving or running or walking or whatever. And actually, these different types of energy, they are not equivalent. So the chemical energy is totipotent, so it can be transformed to other sources. So we can use ATP energy to pump the ions. We can use ATP energy to produce heat. We can use ATP energy to move the muscles and move the animal. But then these others, we can't transform them to chemical energy anymore. So ion currents on the membrane are not affecting directly on chemical reactions, but they are controlling the living or life of the cell. And then if the muscles are moving, they are not producing at the same time any chemical reactions at the moment. So they are not making any enzymes, etc. at the same. And the problem is also that when we are producing heat, there is a not so huge temperature differences that we could use a steam engine or whatever to use this thermal energy. So it's completely useless. And when transforming from one type to another type, it's always a little bit inefficient. So energy can be either useful or useless. So that's why these energy forms are divided in high grade or low grade. And when we are losing energy in low grade, it means that, okay, we are producing heat because we can't use it anymore. And we can be either efficient on this energy transformation or less efficient. And depends just at, okay, what is the output of high grade energy versus the input of high grade energy. And when comparing different kind of processes, we can get some kind of idea on this efficiency. So when we are burning glucose in our cells to produce ATP, the efficiency rate is about 70%. So about 30% is wasted in heat. And so when we are doing from the chemical energy to another kind of chemical energy, that's quite efficient. Then when we are making muscles to move or contract, then when we are turning ATP to this muscular motion, it's 70% is wasted as heat and 30% goes in the movement of the cell. And then when the movement of the cell is moving the bike, even the best bike on the job and summer etc. So there's not many problems with the driving the bike. Even then, the efficiency is about 30%. So we can calculate from this that, okay, when you are burning glucose to move the bike around the campus, about 5% of the energy of the glucose goes in the moving the bike, and 95% goes in the heat. So it's not very efficient. So let's go back on the penguins. When we are eating food, we are ingesting some chemical energy. And some of it will be absorbed depending on what kind of intestine we have and what kind of transporters we have. And then the rest goes in feces. And it contains also some chemical energy. Then this absorbed chemical energy can be used for external work or maintenance or biosynthesis. And the biosynthesis can be used to accumulate to use it to make new proteins, new biomolecules, and etc. So larger tissues, etc. But also the biosynthesis affecting that, okay, some of this chemical energy can be also exported as organic material. So if we have a penguin, they are laying an egg, there's a huge amount of proteins, etc., that they are exporting from the tissue. Then the generation of external work affects that, okay, when they are moving, they are walking or swimming, etc., so they can move around. And that's the way of this external work, how it works. And then the third one is the maintains. That means that, okay, we are maintaining, for example, circulation by pumping in the heart all the time. And all these, at the same time, when we are exporting organic material or building organic material for our own body or moving around, we're also producing heat. So summa summarum, it means that, okay, the energy is entering in chemical form, but then it leaves as heat or chemical energy or external work. And from these, we can get on the metabolic rate. So energy that is converted on heat or the external work, we think that, okay, that's the consumption. And the speed of this consumption is called the metabolic rate or the heat production. And it's important for three simple reasons. First of all, if we know what is the energy consumption, we know that, okay, how much food we need for the animal. If there is not enough food to compare the energy consumption, there is a problem. It also has a quantitative measurement of the total activity of all the physiological mechanisms. So we don't have to worry so much that, okay, what about the leg muscles, or what about the heart rate, what about the kidneys, etc., how much energy they were using in the different kind of processes. We can just put the animal and measure the metabolic rate to have overview of how much it needs the energy. Then the third one is a little bit confusing, because we have also ecological consequences. So depending on how well the animal is consumption, the energy it will get, then it's affected, okay, what kind of relationships there can be in the ecosystem, etc. The metabolic rate can be measured directly or indirectly. In the good old days, we could use these Loventer's apparatus to measure that, okay, the rate at which the heat leaves from the animal body. So there's an animal, and it's surrounded with ice-filled jacket, and around that there is another jacket filled with ice, so there is no environmental heat melting the ice. And then, depending on how much heat is leaving the animal body, then the ice is melting. And by measuring how much water we get, then it depends, we can say that, okay, what is the metabolic rate of the animal. But nowadays, people usually don't have these kind of equipment, but they're using some modern devices. But then, sometimes this is a little bit complicated, so there are also indirect methods to measure by either to see that, okay, what is the respiration gas exchange, or then the chemical energy content. They are cheaper and faster, but then they are a little bit less reliable because they are not testing it directly. So in these indirect calorimeters, most probably gas exchange is the most common. Because the oxygen consumption, we can estimate the metabolic rate from it. So when we are having a test tube with glucose and oxygen, we will get carbon dioxide, water, and quite a lot of energy. And the interesting thing in these chemical energies is that, okay, they are rate independent. So although we are now skipping what happens in the glycolysis, what happens in the citric acid cycle, or in the oxidative phosphorylation, we can get the same amount of energy also in the cellular processes. And still, the problem is that the heat depends on the energy source. So because these different nutrients, they are entering in our energy metabolism in different phases. So carbohydrates are first going on the glycolysis. Lipids and proteins are used in the citric acid cycle, etc. So the heat per oxygen consumption or carbon dioxide production is a little bit varying. But if we have, for example, animal that is using carbohydrates poorly, like the hummingbirds, then we can calculate that, okay, if they consume 10 milliliter per minute oxygen, then the metabolic rate is 211 joule per minute. But the problem actually is that we hardly ever can precisely tell that, okay, what is the diet or what is the energy source, because even we have learned so far that, okay, even in proteins we don't get too many amino acids that we are not using. How to store them? We can cut them in species and store them as glycogen and then use it as a carbohydrates. So then the heat is quite much approximation. And this can be evaluated. There's a quite nice idea of how to evaluate it, because these nutrients are entering in the energy metabolism at different time. So we can just compare how much oxygen was consumed versus how much carbon dioxide was produced. And in theory, if we use carbohydrates, this relation is 1. Then if it's lipids, it's 0.7. If it's proteins, it's 0.8. So we can, if it's something of these, then we can say that, okay, it's using either protein diet or lipid diet, etc. But quite often, if you make this kind of experiment, the value is something between these values. So it's very difficult to predict the energy source. And that's why the researchers are quite often using some kind of approximation that, okay, it's 21.2 joule per milliliter of oxygen for all the calculations. Well, the problem is that because it's approximation, it's causing 5% error. And in physical sciences or chemical sciences, 5% error is quite a lot. But especially when dealing with whole animals, 5% error is minimal. So we don't have to worry on that so much. But the real problem over here is that we assume that, okay, it's always aerobic metabolism that we're using. So if the animal is using also anaerobic metabolism, producing lactate or ethanol, then we have severe problems in this metabolic rate estimation. But even in the extreme in the vertebrates, hypoxia or anoxia tolerance, so the crucian carbs that we have discussed several times during this course, even then it's been estimated that they are using oxygen as long as it exists. So they are using oxidative phosphorylation. So that's why if there's oxygen available, even in crucian carb, we don't have to worry so much on anaerobic metabolism. Then the other way to measure metabolic rate indirectly is to have the balance of materials. So what goes in and what goes out. And this kind of method is widely used in agriculture. And the technique used bomb calorimetry for measuring the energy value of organic material. So you put the organic material in a metal cylinder with quite thick walls, and then it's surrounded with water. And then you practically expose it. There's no large explosion or whatever, but so all the organic material will burn in this equipment and then heat a little bit the surrounding water. And then you can calculate that, okay, how much, what was the temperature difference, and then you know that, okay, how much energy there was. But still, the method assumes that all the energy is consumed in the animals, but still they can actually raise weight. Or they can lose energy also by shredding the feathers or secreting mucus. So if you are sweating a lot, you pee less. So we should measure all the excretion, all the output of organic material. And the problem also is that this will take always 24 hours. So it also only works in long-term measurements of average metabolic rate. So you can't measure every hour of this or whatever, but you get one value after 24-hour measurements. Okay, so we have different kind of ways to measure directly or indirectly. But the problem in all these is that actually the metabolic rate is several factors are affecting on it. First of all, if the animal is moving, so if there's physical activity, its metabolic rate is higher because it's using ATP or chemical other energy, chemical energy for moving the muscles and moving around. Then the environmental temperature can affect. So in the homeotherms, like humans, it's lowest in the thermoneutral zone. So if it's warm, that's not too cold or not too hot, then our metabolic rate is lowest one. But then if in sauna or when winter outside, it's always higher. And then in poikilotherms, for example, it's higher in hot and lower in cold. So because they don't have any thermoneutral zones, because they are the body temperature is varying all the time. Then, if we have a meal, it will rise the metabolic rate for several hours. We will discuss a little bit later this. Then the body size is affecting. So the rate per gram of your tissue is increasing when the body size is decreasing. Then the age is affecting. So in humans, for example, it's higher in the puberty. So when you are a young adult, but then it's decreasing. And the gender is affecting. And there's also a species-dependent variation, but in humans, in the males, it's higher. Then the oxygen concentration can affect. The low oxygen concentration, then it's lower metabolic rate. But if it's enough or too high oxygen concentration, it doesn't matter anymore. Also, the hormonal status can affect. That okay, because we have thyroid hormone that is affecting on the metabolic activity of certain processes, so if the thyroid secretion is increasing it in mammals. And also the time of the day is affecting. So because humans, we are day active animals, then the metabolic rate is also higher. And last but not least, also the salinity of water in aquatic animals is affecting. Especially on marine crab, this has been tested that okay, when they are put in low salt environment, they need more energy to maintain the salt inside them. So there are different kind of factors affecting on the metabolic rate, and that's affect that it's quite problem or difficult to measure it right. And one example is the meal. After having a large meal, you get tired, and it's based on this kind of dietary induced thermogenesis or specific dynamic action. And it just means that okay, there's a temporary rise in the metabolic rate after feeding. And it's actually quite large rise. So it affects on how much energy there exists in the food. And so in experiment when they were testing in fish and getting it 1.5 kilojoule or 5.6 kilojoule, you can see that the oxygen consumption is about 2 times, a little bit more in the higher energetic meal, but it's also longer, and it can last for days. And it's been estimated that about 10% of the energetic intake of human meal is goes in this dietary induced thermogenetics. But then if we are having protein rich diet, the rise can be over 30%. And that's why it's being quite often speculated that, okay, if we could find some food material that we can't ingest it, or so we can't absorb it in our circulation, but if it works in adding this thermogenetics, that would be a very good way to get thin. But still, the mechanism behind this phenomenon is not known. Okay, because the metabolic activity is affected by several different factors, then the problem is that we should have some kind of standardized way to measure it. Because otherwise it's completely impossible to compare that, okay, is human consuming more energy than mouse, etc. And in the clinical studies, it's quite simple. So 12-hour fasting is essential. But then depending on vertebrate or on homothermic or ectothermic animals, we have different kind of metabolic rate. We either have basal metabolic rates on birds and mammals, so they must be in thermoneutral zone, not too cold or not too hot, and fasting and resting. And then we have standard metabolic rate on the ectotherms. Because there's no thermoneutral zone, then they have to have fasting and resting. And for every animal species, in these cases, there's a lot of different kind of standard metabolic rates in the tables, because they've been tested in different temperatures. And in both cases, they are not eating and they are resting. But sometimes the resting is quite difficult to identify. Thank you.